For our final results, in the control group, we observed a maximum growth of 0 centimeters in the 50-50 regolith potting soil mixture and a 19 centimeter for the maximum growth for the 75-25 regolith to potting soil mixture. Conversely, the experimental group, we observed a maximum growth of 23.5 centimeters in the 50-50 regolith to additive mixture, a 13 centimeter maximum growth in the 75-25 regolith to additive mixture. Taking the average of these maximum growths and the plant number 2 growths, we observe that the additive growth medium overall outperforms the generic potting soil. Looking further, however, we can also observe that the 50-50 additive mixture supported a greater growth compared to the 75-25 additive mixture. From this, we can conclude that the 50-50 additive mixture is the optimal growth medium and lunar additive ratio for growing these cowpeas.
To ensure that our experiment was both replicable and feasible, we used standard 300 milliliter glass beakers as our planting containers, and each beaker was layered with the appropriate growth medium. 2 Vigna unguiculata seeds were labeled Seed A and Seed B, and they were planted in a hydrogel layer within each pot to reduce the risk of seed failure. Each planted beaker was connected to a 300 milliliter external water reservoir through a wick system, and this allowed water to be absorbed gradually by capillary action, maintaining constant soil moisture throughout the entire experiment. This setup was replicated across 5 pots, each representing a different substrate type or regolith mixture ratio, and all pots were placed together under a grow light and a single fume hood to ensure identical environmental conditions.
To get the most accurate results, consistency was our top priority. We know that too many moving parts can make data hard to read, so we kept these 4 factors exactly the same across every single trial. Water. Every reservoir received the same amount of distilled water. Environment. All plants were kept under the same fume hood. Quantity. We placed exactly 2 seeds in every pot. In species, we used the same type of plant for the entire study. Now, for the variables we actually changed. Our primary focus was the additive specifically made for lunar regolith. Our control group used standard potting soil, and our experimental group used a mixture with the lunar regolith additive. But we didn't stop there. We also tested the ratio. By changing the percent growth medium in the soil, we could determine the optimal balance between the regolith and the additives. Regarding our dependent variables, we tracked the growth through 2 lenses. First, the quantitative data. We took data on stem height and leaf length, the total number of leaves, and soil water saturation, tracking exactly how much water the system absorbed. Second, we looked at the qualitative data. By observing leaf coloration and overall health, we gained a deeper insight into which growth medium truly performed the
Some advice to future participants is to standardize the way you take data so that the data is accurate and consistent. Also, before writing the final report, have an idea of which graphs to create.
We are the Sprayberry STEM Moon Explorers Team 2, and we have planted the moon.